Today's lesson takes us back to 1953 when two microbiologists proposed that our DNA structure was a double helix. This zoomed in is going to look like this. Our double helix is composed of a phosphate backbone with this formula. It's negatively charged, and remember that for later. And the base stacking of the double helix has complementary pairs with our nucleobases. So adenine pairs with thymine, cytosine pairs with guanine, and they have a hydrogen bond between them. The miniature arrow and major arrow represent major groove and minor groove, and these are just binding sites for proteins. A normal DNA strand is going to go from 5' to 3', and its complementary strand is going to move from 3' to 5', which is why they're considered anti-parallel. We can break up our nitrogenous bases into two more groups, a pyrimidine and a purine. So you'd say, well, these are complementary bases, so how come they're not the same? Well, they're different in structure. Thymine is going to have a similar structure to cytosine and guanine with adenine. Thymine has a 6-member structure, as seen below, and adenine has a 5-member and 6-member structure, and that's going to look similar to guanine. There's one more pyrimidine I didn't mention, and it's uracil. The RNA doesn't contain thymine, but it still has to have a complementary base with adenine, so uracil would take thymine's place. Next thing we're going to look at is handedness. So like people write with their right or left hand, DNA double helix can move in one of two directions. So for me to distinguish amongst the two, what I do is I take a portion of the DNA and I look at the base and I say, well, is it hooking right or is it hooking left? And that seems to work for me, but if it doesn't for you, there are many other ways to look at it. Next thing we're going to look at is our sugar structures. So we have ribose and deoxyribose. And they look the exact same, except at the 2' location, deoxyribose doesn't have an oxygen, which is the main differentiator between the two. Next thing we're going to look at is our nucleotide. We have our phosphate group, we have our nitrogenous base, and we have our sugar. This is not a nucleoside because the nucleoside would not have this phosphate group. The ribose carbons have several functions. So the 1' function is to bond to our nitrogenous base, in this case guanine. Our 2' differentiates between the sugar molecule we're using. The 3' is dealt with in gene expression, and 5' binds to our phosphate group. The last thing we're going to look at is our temperature of melting, which is the point at which 50% of the double helix has changed to a single strand. Remember when I said before that our phosphate backbone had this formula and it was negatively charged? Well, if we ignore the genetics for a second and look at the chemistry, these negatively charged particles are going to repulse off of each other because they want to get as far away from each other as possible. They're not attracted to each other. They would rather have a positively charged particle. If we add a salt concentration with sodium plus, which is positively charged, if we start throwing positively charged particles into here, they're going to start binding instead of repulsing off of each other. So before, with the negatively charged particles, the repulsion was increased, which increased the kinetic energy and sped up the temperature of melting. But if they start binding, the repulsion goes down, the kinetic energy goes down, and the temperature of melting would decrease. Thank you for watching. Here are my references.